Welcome to Nikolay's genetics lessons, and here's a question I got from one of my subscribers. Why bands are stuck near the well in agarose gel electrophoresis? And this is exactly what you see on this picture. The ladder went all the way down, but bands which you see here in black color, most of them, some of them also went some way down, but most of them are stuck near the wells. So here is the message. I'm running a gel 0.8% agarose gel with my PCR product. In the gel image I can see some black bands stuck near the well. Is that my amplicon or some other thing? What all different things I can try to rectify this? Well, let's take a better look at this picture. There are three main culprits behind this common lab headache. The first one is heavy genomic DNA. If you use too much template, large genomic DNA is typically too bulky to squeeze into the gel matrix. Second reason for a picture like this: protein-DNA complexes. Enzymes like Taq polymerase can stay tightly bound to your DNA, creating a massive slow-moving aggregate. Third reason is insoluble gunk. Leftover cell debris or polysaccharides from your extraction can trap your amplicon in sticky mesh. And here is how you can fix it in your next run. Add SDS to your loading die. A tiny amount of detergent of 0.1% to 0.5% of SDS will strip those protein right off your DNA. My second advice would be dilute your template. If you're starting with genomic DNA, dilute it 1 to 10 or 1 to 100 before PCR. Less is often more. And the last advice would be do a spin column cleanup. Running your PCR products through a silica column removes all proteins and debris, leaving you with clean, last running bands. So try these advices and watch your DNA run perfectly. And this is all for today. Subscribe and see you in the next video. Goodbye.